Hey, super quick today. Today we'll be going over the physiology of smooth muscles. So, remember in histology, you learned that smooth muscle is fusiform and the cytoplasm is granular without any striations or sarcomeres, and there are more thick than thin, there's more thin, than thick filaments, and they all kind of like link to the dense body in a diamond mesh like pattern in order for it to contract. It doesn't contract in one big unit like skeletal muscle. Instead, it contracts and bunches up. So there is some tropomyosin, but there is no troponin. So there's not going to be the protein that blocks the tropomyosin. Instead, there's going to be calmodulin, caldesmin, and calponin, which kind of sort of does the same thing, but they really regulate the smooth muscle contraction. And the other difference is that the smooth muscles don't have transverse tubules like invaginating in to make the entire muscle contract as one unit. Instead, they have calveolae, which are not like direct invaginations. They're just kind of like pits that come in. The calveolae really is there for kind of like receptor binding rather than actual contraction. So in the smooth muscle, it's considered a single unit because all of the cells work together as one, and they are in these systems, and they're connected to each other via the gap junctions rather than the T tubules in order to communicate. There are some multi-unit smooth muscles where all the cells contract individually. Usually that's in the eye. All of those are very specific, very specialized cells, so they're all innervated separately. So in terms of contraction, there isn't any neuromuscular junctions compared to the skeletal muscles. Instead, they're varicosities, which release the neurotransmitters. So the varicosities are sort of like axon terminals. They kind of have the same process, like calcium will enter, but instead of binding to troponin, it will bind to calmodulin. So it's similar. The steps are similar, but there are different proteins and different fibers in smooth muscles. So unlike skeletal muscle, the activated calmodulin will activate something called the myosin light chain kinase. So the myosin light chain kinase will transfer a phosphate to the myosin, activating the myosin ATPase, and then the activated myosin will form the cross bridge, just like in skeletal muscle. But instead, remember that they use calmodulin and the myosin light chain kinase. So as you can see here, there's a couple of different, proteases and enzymes that are at work. So the myosin light chain phosphatase comes in, rho kinase will dephosphorylate it, and then the myosin heavy chain gets phosphorylated and then is activated, then it can bind. So there is the myosin binding domain of the myosin light chain kinase, and there it needs the calmodulin and calcium to bind, like here, in order for this to be dephosphorylated in order to phosphorylate the myosin light chain. And all of these really require calmodulin. Okay, so the trigger, instead of an action potential, is a rise in intracellular calcium levels. A subthreshold depolarization can open the voltage gated calcium channels, kind of stimulating the contraction, but it's not necessarily an action potential because the calcium can enter from this extracellular space and the sarcoplasmic reticulum. So instead of being very fast and expensive energy wise, smooth muscle is slow, and the latent period in the skeletal muscle is about 2 milliseconds, while in smooth muscle is about 50 to 100 milliseconds, so significantly longer. The smooth muscle stretches more and generates more tension when stretched than skeletal muscle due to the cross links of the dense bodies and the thin filaments. So calcium will come and bind to calmodulin, and that initiates a cascade that essentially ends with the phosphorylation of the myosin light chain and the activation of ATPase. So as long as the calcium is present, the myosin light chain stays phosphorylated, so the myosin head can repeatedly bind to the ATP and continue the cross-bridge cycles. That's why it's like kind of like rhythmic pulsations in the intestines. So like it's not like one unit contracts, like this end might contract while this end might not. It's really used for the peristaltic movement. So instead of having summation of action potentials, the force of contraction increases with just increasing the calcium influx. So the higher the calcium concentration, the stronger the contraction, only in the smooth muscle. So without calcium calponin or caldesmon to bind actin and inhibit the myosin ATPase, that prevents like the cross bridge formation. So here's the mechanism. So the stimulus comes, calcium comes in, and then myosin binds. ATP is hydrolyzed in order for this to be released. The myosin head is released, and then the ATP, right here, ATP is present, ATP binds to myosin, is released. Skipped ahead a little bit, and then returns to the starting position, and then troponin will bind, will release the calcium, so it needs another calcium to bind in order to start the process again. So once the calcium levels go down, it's reset, and then you need more calcium in order for the cycle to start again. So calcium goes into the cells via the voltage-gated calcium channels, which are the IP3 gated channels. As you can see here, calcium comes in, and then it can turn to the phosphoproteins, and then it can also come in through here when a ligand binds, and then there's the G protein shuttle, and then IP3, and then you pump out the calcium into the endoplasmic reticular membrane, and then from there it goes out and goes and makes the photoproteins. They are two different kinds of inhibition. One is voltage independent and one is voltage dependent. There are multiple mechanisms that include the trafficking and the signaling pathway, along with phosphorylation in the voltage independent path, while in the voltage dependent path there's only one mechanism. It's just the direct binding of the glycoprotein to the subunit of the channel, and then bam, allows the calcium to come in. So during relaxation, there's going to be a reduced release of calcium, of course, and that in turn reduces the phosphorylation of the myosin light chain and the dephosphorylation of the myosin light kinase, like the ones that already are phosphorylated. So the reduced calcium entry will cause the calcium ions to be unbound from the calmodulin, and then the myosin phosphatase dephosphorylates the myosin light chain, and then finally the smooth muscle relaxes. Calcium is reuptaken by the circa and is resequestered into the sarcoplasmic reticulum. So as you can see here, so the L-type calcium channel allows the calcium to come in. Calcium does its job. There's a contraction there, and then once we are good to go, like when nitric oxide comes in using the GMP cycle, or Rho kinase comes in to inhibit the contraction. The calcium is released and then is unbound from the calmodulin. Okay, that's it for now. Thank you for your attention and good luck studying.